Okay, we have a class today. It's important for the review of our exam 3. Exam 3 will be on Thursday right here. Okay, Carly is here. She already have a tutoring section last night, and tonight 8 o'clock in the lab room, she will have another additional review section if you need it. And after class, I will post posted all the key to the eCampus. Okay. You can go over all these questions, but I want to make sure there are some key important things we need to know. The first question is question 18, the calculation of the ATP. So during the aerobic respiration, and I mentioned that we have extra points, which means you know the aerobic respiration cycle of the chemicals and also the structure names. But that's extra points, which means you don't have to do it. If you are only in here, you never learned biochemistry, genetics, and you said, I only need to be doing the work what required here, then I'll tell you an easy way to do, okay? So let's say calculation. That's called the calculation of ATPs during aerobic respiration. Okay, how are we going to do? We skip all the chemicals. You know glucose. Okay, glucose goes here, have pyruvate. And this is called glycolysis. This is you need to know. If you don't know this, then there's a big problem. Okay? Glycolysis. I don't talk about anything else. So how many ATP generate? And then there's a transition step, become acyl-CoA, and then you have this cycle called the TCA cycle. Okay? Now let's talk about these one by one. Okay, let's go here. Glucose goes this guy, pyruvate. How many ATPs they do at the top? Hydrolyze 2 ATPs. Okay, that's simple. How many ATP happen here? How many? There is one NAD reduced to NADH2. Is that right? Then what happened here? There is one ADP becomes ATP somewhere here. Then finally, when phosphoenolpyruvate becomes pyruvate, there is ADP becomes ATP. So I don't even know exactly where it is. So how many ATP generate here? It's 2 multiplied by 2 equals 4 ATPs. And what you learned in your biochemistry class, so total is 2 ATPs for glycolysis. But I said, because this is part of the aerobic respiration, so this NAD reduced to NADH2 generates 3 ATPs. Okay, and then multiply by 2. Is that right? So how many? 6 ATPs. Okay, here is something I want to be clear. NAD reduced to NADH2, what I do is 3 ATPs. You can do 2.5. It's up to you. Then FAD reduced to FADH2 is 2 ATPs because it's easy for calculation. You can do 1.5. But you have to using only one set. Okay, not like a mix. So how many here? -2 plus 6 plus 4, that's 8 ATPs. Okay, that's finished the glycolysis. Now we're going to talk about this guy, from here to here. Again, I don't know all the chemicals, so pyruvate, go acyl-CoA. That's called the transition step, then we do the cycle. Okay, I don't know anything, but I know NAD reduced to NADH2 right here. There is NAD reduced to NADH2 right here. There is another NAD reduced to NADH2 following that. And somewhere here, I know there is a FAD reduced to FADH2. And somewhere on the top, there is NAD reduced to NADH2. Okay, don't forget, there is another GDP will generate. So GDP becomes GTP. And this is equivalent to ATP. Okay, then you do the calculation. This is 3, this is 3, that's 3, this is 1, this is 2, this is 3. So how many is 3? 3 multiplied by 4, add 2, add 1, that equals 15. Because one pyruvate is one cycle, another pyruvate another cycle, so we have to multiply by 2. That equals 30 ATPs. And then you add up here the 8. So 8 add 30 equals 38 ATPs. This is a requirement for this class, which means you don't need extra points. This is certainly answers the question of 18. Now I want to make your life easy, so I already prepared. You always need to go back to look at the eCampus. I have a picture for you. So you can use this one. That is the detail of everything. Okay? Look at here. Everything is there. I took a picture. Which means there's a chemical name, there's a structure name. If you need extra points, that will something you can use. Now, there is one thing I want to mention is citric acid cycle. Citric acid become isocitric acid. We don't have cis ascorbic acids for bacteria. But for the fungi, you have it. That's the only difference. Okay, is that clear? This one will help you. It's up to you. If you think it's an overload for you to memorize everything, but you at least need to write some glucose. Like write pyruvate. Like write glucose 6-phosphate, like citric acid. That more or less give you some extra point. Is that right? So that certainly it helps you. Okay, so this is the first thing I want to talk about that. Okay, second thing I want to talk about is, so I keep it a little bit, you can take a notes, is the lacto-operant. Because I said this is an important topic for the exam. Everybody should know. And there is a different situation. I know some of you looks like it's a maze. Because there's a 4 different situations, and we have 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. Okay, so we have cap, promoter, operator, lac Z, lac Y, lac A, so CPO, and we have a lac Z, lac Y, and lac A. So we have a CPO. You can draw like this, okay? Z, lac Y, lac A, CPO, lac Z, lac Y, and lac A. Okay, we have a scenario. Because the lacto-operant is based on this curve, they use glucose first, and then when it's done, they use lactose. It's called dioxyriboose. Okay, so the first scenario, there is glucose, and there is lactose. Is the transcription will happen? It's very simple. Is the transcription going to attach to promoter or in a polymerase? That's the only answer we needed. Okay, yes or no? Glucose and the lactose, we mentioned This is a dual channel control, which is negative control and positive control. Positive control is CAMP, C-A-M-P, and this is, we call it a tetramer inhibitor. And sometimes you call it like a repressor. It's the same meaning. So if there is a glucose and a lactose, this will be not attached, but cap also not attached. So no. Okay, the second scenario. There is no glucose, but with lactose. Is the transcription will happen? Yes. Because cap will be there, and this will come. Is that right? Okay, third scenario, there is no glucose, and there is no lactose. Well, this is easy. Of course, nothing going to happen. You don't have anything. You only have distilled water. Last scenario, there is glucose, but no lactose. Is the transcription going to happen? Of course not, because this inhibitor will be there. Okay, that's a 4 different scenario. You should be well known, because we have some questions we'll be asked you about that. Okay, so that's another big topic. Another topic I want to mention here is this question. It's question number 12, because this question is important. We talk about at the beginning of the fermentation. A very typical formula for fermentation and for homofermentation. So pyruvic acids will become lactic acid, and NADH to reduce to NAD plus. Okay, so how are you going to answer that question? When you see NADH2 become NAD plus, I said this is like 2 empty vessels. You could add H there later on. So what this means? The electron, which means the proton is gone. This is certainly is oxidized. Is that right? So what we call pyruvic acids reduced to lactic acid. That's the first question. So let's say you have this 12 question. Name this type of biochemical reaction by the microorganism. Well, it's fermentation. Okay, this is simple. This is homo fermentation. And you can just say it like this, homo fermentation. Name the reduced products. This is reduced products, lactic acid. So NAD is what? Oxidized product. That's NAD plus. What is reducing agent? NADH. What is oxidizing agent? Pyruvic acids. Okay, so that's the answer. What is the biological function of NAD plus in this reaction? Electron carrier. Okay, NAD plus. This is electron carrier. And I said, very important, this has to be recycled. The reason why fermentation only 2 ATPs generate, but the bacteria still can survive, because it is coupled with glycolysis. So when you learn in your biochemistry class, they say the glycolysis only generates 2 ATPs. We assuming it is connected to fermentation. But when we here talk about aerobic respiration, this guy you have to consider it. Okay? So that's the thing. Name the food product processed by this reaction. Well, there's yogurts and all kinds of things you can can any fermented food products, but not wine, not vinegar. Okay, because those are hetero-fermentation. Generally, the alcohol is different. Vinegar is secondary, secondary fermentation. That's different. Name the microorganism related to that, lactobacillus. So that's just a question we want to talk. Okay, these are the things that are important. So let's go over some questions. You don't have to worry the first one. The first one will be tested in the final exam because we're going to talk about safety. US EPA, well, you read the news, EPA may be gone. Okay, so we'll see. So those are the questions we're going to test you later on. FSI, FDA, we'll talk about that exams 4, final exam. Okay, let's look at these questions. A non-protein component of an enzyme that is loosely attached to the protein is referred as, if it's loosely attached to what it is that called a coenzyme, is that right? If it's closely attached, we call it a prosthetic group. So, this is something we talk about. So we have the enzyme, and most of the time we call it a hollow enzyme. It has a protein factor, we call it a cofactor. Then we call it a coenzyme. Coenzyme usually is loosely attached. If it's closely attached, it is called prosthetic group. And we have that one to talk about. Then we definitely talk about RNA polymerase. We talk about alpha 2, beta, beta prime, omega, and sigma factor. And this is called coenzyme. Sorry, this is called a coenzyme. Hollow enzyme, we have a coenzyme and a coenzyme, cofactor. Okay, coenzyme. This is called loosely attached, we call it a cofactor. Closely attached prosthetic group, this is called a coenzyme. Okay. So that's coenzyme. Because in such a way that disorder increases, that's a second thermodynamic. This question. There is a cell bacillus, make energy from oxygen and iron. Then we have the voltage value. And we have those chemicals, so how we do it? This is something we talk about in the lecture 2. Electron is always from donor to acceptor. How do you know it's a donor? We're using 2-half reaction, test the voltage value. It is going to be more negative. And how do you know it's an acceptor? It is more positive. So what's going to be the flow chart here? Which one will be on the top? The smallest one. So Fe2 will be on the top. Then go second one. Because this is 0.77. What are going to be the second one? Lustothiamine. Cyanine will be the second one. That's 0.78. Okay, the next one. Cytochrome C. Then cytochrome A. And the last one is water. Is that right? So it go this way. So that's 0.79, then 0.8, and last one 0.82. So the electron goes this way. because at the beginning the voltage value is smaller, more potentially electron donor, and always oxygen at the back, at the last, because it is a more potential electron acceptor. Okay, so that's the easiest way to do. If you want to go back to study the slides, this will be correlated to, this is I always want you guys to do, is go back to read your slides also. You could see is these slides. The greater, you see, the more negative on the top, the more positive at the bottom. This is a two-half reaction. Okay, these couple of slides, these three slides related to this question. Okay, so you have to go back to read the slide, look at the slides all the time. Which of the following, question number 5, which of the following process generates the greatest amount of the energy? Fermentation only 2. Substrate level phosphorylation. Well, that's a name. How they generate energy. Opposite to oxidative phosphorylation. That's not, we are talking about how much energy they're going to generate. Anabolism, which is don't create energy, they use energy. Glycolysis is only 2. Anaerobic respiration is somehow between 2 and 38. Now, how are you going to find the answer? This is the one I said that the very important slides right here. You should absolutely read, which is all the way here. This is a very good conclusion slide, which tells you, comparing aerobic respiration, anaerobic respiration, and fermentation, how many ATP they generate. This is tells you anaerobic respiration is 36 or less. Okay, that's why it's more than fermentation. So if you want to go back to see the slides, this slide is important. Sulfur drug or antibiotics that function by inhibit synthesis of vitamin by competing the normal structure of the activity site of enzyme involved in the vitamin synthesis. So that is going to be C. This is the one we go back talk about the enzyme. You should know that's what the sulfur drug looks like. Because the sulfur drug is a very similar morphology to the beta-benzoic acids. Beta-benzoic acids is a precursor material to generate folic acids. Folic acids, bacteria cannot create by themselves, has to come in from the nutrients. So you can see the picture here, which is explain well what is the competitive inhibition. And this you should be very familiar because it talks about about chemistry all the time. Question number 7. The transfer of electrons and hydrogen by a dehydrogenase to an organic molecule as the terminal electron acceptor is called A, B, C, D, E, F, G. This is kind of confusing you. But you should always remember at the beginning of the class, we talk about the fermentation, aerobic respiration. This is something you should know. Electron donor, electron acceptor. If the electron donor is organic chemical, electron acceptor is also organic chemical. This is called fermentation. If the electron donor is organic chemical and the electron acceptor is inorganic chemical, and this is called respiration. And the respiration have 2 different categories. If this is oxygen, the final electron acceptor, then we call it aerobic respiration. If it's others, then anaerobic respiration. So that's what the picture looks like. And I said lots of the times, this is the best definition to talk about fermentation. which is electron donor is an organic chemical and the electron acceptor is also organic chemical. Therefore, you can see here the electron donor is pyruvic acid and the electron acceptor is lactic acid. Okay, that's well explained about that. So the answer here is to an organic chemical. This is called fermentation. What is the best definition of fermentation? Well, that is easy. So what is called talked? The oxidation of a carbohydrate with organic molecules serving as a final electron acceptor. So that's C. Which of the following is used electron carrier by leading organism of the choices? We have a slides for that. Slides right here. Carriers, oxidized form, reduced form, electron carrier. Okay, this slide is important, you should review. When the end product of a pathway inhibits catalysis of the first step, that pathway, this phenomenon is called, that should be called feedback inhibition. We have slides for that. Feedback inhibition. Can you see it? That end products affects in the subterminal and the terminal substrate. So you can understanding if you generate too much pyruvate, then glucose 1,6-biphosphate will be inhibited and the glucose will be inhibited. It's the same thing, okay? Then you go here. We don't have to do 11. This we talked about already. You don't have to do 13. Okay, 14. Which of the following can be used as electron acceptor during anaerobic respiration? Well, the inorganic chemical other than oxygen. So all of them. Where are the slides? The slides you should be looked for is right here. Anaerobic respiration. That's number 1. Use the electron carrier other than oxygen, and this is examples. Anaerobic electron acceptor is oxygen. Anaerobic is nitrogen, sulfide, carbon dioxide, ferric acid, all the others. Question number 15. Which of the following is not true regarding ATP synthesis? They require promoter to make ATP. They expand the inner membrane of the mitochondria. The proton flow is outward. Well, this is test you what you should have very clear. This is test you. What is chemosmosis? Peter Mitchell in 1929 pointed out, this is win the Nobel Prize later on, the chemosmosis theory which related to ATP synthesis. So what happened? Electron transfer, then they repel outside of the cell membrane accumulated based on the dehydrogenase. Then lots of the proton accumulate there, become a potential energy called a proton motive force. And once it's accumulated more and more, they will be activate ATP synthesis, so ADP becomes ATP. At the same time, proton will go back, triggered, coupled with flagellar rotation. And we mentioned about flagella, we have a Petrotrichus, which is Proteus bulgarius, is that right? That's why we use SIM test to can identify that. So this is a very interesting theory related to what we learned. When you talk about this way in the biochemistry class, you may confusing, but when we talk about in the bacteria, you know we talk about the flagella. It is related to that. So the knowledge will be automatically connected with each other. Okay, so that's 15. The hypothesis that proton motive force driven the ATP synthesis is called, right on the top, chemosmosis. So these are the slides you should always go back to read when you do the review these questions. Organisms that form lactate, ethanol, carbon dioxide as end products, that is called heterofermenter. So this is what we call it a hormone fermenter. If you generate alcohol, like you make a wine, that is heterofermenter. And where you can find the answer, you should always go back and we talk about the fermentation, top Pyruvate become lactic acid. This is hormone fermentation. Bottom pyruvate become acetaldehyde, then become alcohol. This is heterofermentation. So these are the two examples. And this slide tells you they coupled with glycolysis, so they only generate 2 ATPs through substrate level phosphorylation. And this is the one showing you the fermentation products. So these 3 slides, you could be put them together. And on the top here, you will see the definition of fermentation and the respiration. So these couple of slides, you could be connected together to study. Okay, question 19. Well, this is don't let you guess because we go all the way to K. If yeast metabolized 1 molecule of glucose to ethyl alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? There's only 2. Fermentation only generates 2. It doesn't matter how complicated it is. How are aerobic respiration and anaerobic respiration similar? We already talked about this. How are they similar? The final electron acceptor is inorganic chemical. So you can find here, both use oxygen as a terminal electron carrier. Wrong. Only aerobic respiration. These are other names of glycolysis. Of course, it's wrong. Both utilize inorganic molecule as terminal electron acceptor. So C is correct. Okay, you don't have to read the others. Probionic acids make probionic acid, acetic acid, carbon dioxide in the Swiss cheese. These products result from anaerobic respiration, substrate level phosphorylation, fermentation, and the product of repression. Carbon fixing, where they come from? Of course, comes from fermentation. This is where it comes from. We talk about the products. Probionic acids, carbon dioxide, probionic acids for Swiss cheese, and those wells, those holes, which is for generate from carbon dioxide. Okay, so that's a question from there. Okay, what's the name of this cycle? Yeah, that is called citric acids. Are there oxidative phosphorylation in this cycle? Yes, where are they? Oh, I take it off. Oh, I didn't take it off, so I just need the other one. Where are substrate level phosphorylation? Where oxidative phosphorylation? Whenever you see NAD reduced to NADH2, right here, right here, right here, and here, this is oxidative phosphorylation. GDP reduced to GDP generates GTP. This is substrate level phosphorylation. So we answer 1 and 2. Label the place where so-called transition step. Transition step is right here on the top. Pyruvate to become acetyl-CoA. Where are the slides? If you doesn't know about it, we have slides for that. This is a transition step. Pyruvic acid become acetyl-CoA. This is a transition step. Okay. You don't have to worry about this one. Metabolic precursor, I didn't talk about that. You learn in your biochemistry class, like alpha ketoglutarate, all those are some of them are metabolic precursor. Okay, so you don't have to worry about this one. I didn't mention in this semester. In aerobic conditions, question number 23. It takes sugar to produce the same amount of the ATP when compared to the anaerobic respiration, more or less. Is that less? Yeah, less amount of the ATP to produce it when compared to anaerobic. Okay, which of the following can be used as an electron acceptor during anaerobic respiration? We mentioned already, it's ABC. Central metabolic pathway, I didn't mention in the classroom, but it's a fermentation, glycolysis, and citric acid cycle. Distinguish between first and second dynamic law. You can always go back to see the slides right here on the top. What is the difference between A and first and second? First is energy never created or destroyed. Second is always disorder is increased, which is entropy is increased. Now, by the way, this is extra points, 5. Okay, for adenosine 5 prime triphosphate. So the ATP structure will be extra points. Okay, let's go to C. Name one final product generated from pentose phosphate pathway. Well, let's look at this. What is the answer for that? I said I didn't talk about this too much. What are the final products? 5-carbon sugar, ribose, biosynthesis of DNA and RNA. Okay, you can just simply say it's a 5-carbon sugar. Okay, that is done. So this question is done. Now we're going to look at the second set of the questions. Question number 2. This is basically is genetics. So we want to go over these one by one. 2 strands of a DNA molecule. They are oriented in opposite direction. What we call? Yeah, anti-parallel. Is that right? A consent sequence in the untranslated leader sequence of a prokaryote mRNA, which serves as a ribosome binding site, is called. How we call it? SD sequence? Is that right? Yeah? That's called the SD sequence, which serves as a amino acidyl tRNA synthesis categorized combination of 2 mRNA. FMET. So this one, we need to go to see the slides. We're going to look at the slides. Will be right here. This is we talk about the translation, transcription. Not go to the translation yet. The translation will be right here. So if you forget about that, which one will be generated? That's FMET, N-formyl-methyl tRNA. Okay. The genetic information in DNA is divided into units called. It should be called what? Genotype or genes? Genes. The basic difference between DNA and RNA rather in sugar and the pyrimidine basis. RNA has ribose and the uracil, whereas DNA has deoxyribose and thymine. That's correct. Which is not a pyrimidine. So we have a purine. Adenine is purine. Who else? Thymine. Adenine is not a pyrimidine. Is that right? It's a purine. Cytosine, thymine, uracil, these are pyrimidine. Adenine is a purine. Which one is correct? Which one is not correct? A and T, G and C. Now, that's a tricky thing in the DNA molecular. Uracil is RNA, so C is wrong. C is correct. Okay. Which of the following is true about structure of DNA? Purine, pyrimidine, attach 1 carbon. Is that good to carbon number 1? Yes. We draw the structure all the time. So we draw the structure all the time. So here, this is base. Then you have H. This is going to be OH, OH. This is H, OH. You have H, CH2. You go here, you have O, phosphate. Is that right? That's all we have. Then you have an ATP, just go. So that's correct. Purine are joined by phosphodiester bond. Yes, it's the phosphodiester bond. Remember, you go here. How you gonna connect it? Phosphodiester bond. Is that right? You connect it with a CH2, you go phosphates, phosphodiester bond. Adjacent bases are stacked on top of each other. One pair is 0.32 nanometer. Yes. So all of the choices. So if you're not familiar with this, we go back to see the slides. Will be the DNA structure we talk about right here. Phosphate, sugar phosphate. This diester bond is right here. Okay? Glycosidic bonds right here, diester bond is right here. So that's the slides.